And welcome back. This video is going to be the second half of the Chapter 1 lecture review from Robbins, and we're pretty much going to pick up right where we left off, which is covering the cell following the genome. So the cell itself has to be able to execute a bunch of basic functions just in order to stay alive. It needs, first of all, protection from the outside environment, so that's everything surrounding the cell within your body. It also needs some way to acquire nutrients. It has to be able to talk to other cells. It needs to be able to move around. It needs to be able to generate energy, and a lot of that energy generation comes from the breakdown or catabolism of larger molecules. And it also needs to be able to renew older molecules that are basically old and worn out. So how it goes about doing a bunch of these things comes first of all from the outer shell or the membrane itself that surrounds the cell. So the cell membrane it has a bunch of basic functions, but it's going to do things like providing protection because it is a phospholipid bilayer, but it isn't just a static kind of covering for the cells. It's actually an active surface that is going to choose what can go into the cell and out of the cell. Some of that will be passive. Things like water, oxygen, and carbon dioxide can diffuse passively across the cell membrane, while other molecules like glucose, due to their size, can't go through the cell membrane, and likewise with ions, and that is due to their charge. They won't just passively diffuse through the cell membrane, but the cell has a way to move those actively from the inside to the outside of the cell. Now, we did just say that those ions can't be passively moved through, and the cell actually uses this to its advantage. So we just need to remember that water will follow solutes, so the sodium and the chloride, either in or out of the cells depending on the solute concentrations. Now, extracellular salt, so salt outside of the cell, above what is in the cytosol, that is hypertonicity, so water will leave the cell. The cell will actually shrink because of that, because it's following the salt outside of the cell. If there is hypotonicity, so if there's more salt within the cell than the extracellular environment, water will actually flow into the cells. Now, because the cell itself, the cytosol, which is the fluid within the cell, has a bunch of charged metabolites and of these solutes and proteins that are going to increase the amount of kind of water attraction into the cell actually has to constantly pump out those ions and pump out those molecules. Otherwise, they're just going to fill up like a huge balloon and eventually burst. So that's always kind of an active process that takes energy. But when a cell gets to a point where it can no longer generate energy, which is usually because of some kind of harmful response, it's actually going to swell and swell and swell, and eventually that is going to cause the cell to rupture. Now, how the cells actually stay linked and attached to each other is through actual physical links that are quite small, but it holds these cells together. And there are three main types that we can see here. The communicating junctions, these are also known as gap junctions, and these will actually allow the movement of chemical or electrical signals from one cell to another. And this is important when a lot of cells need to act together at the same time. So something like cardiac muscle cells, they'll need to contract together in a rhythm, and they actually have these gap junctions present, so that as soon as some depolarization occurs, it can flow as a wave through the entire heart, and that gives you efficient contraction for blood flow. These anchoring junctions, also called desmosomes, which we can see in the middle here, this is what is going to anchor cells and the intracellular cytoskeleton to other cells or to the ECM, or the extracellular matrix. Now, when this attaches cells to the ECM, those are called hemidesmosomes, and when the cells are attached at a single point between each other, those are called spot desmosomes or immaculate adherens. And broad bands of attachments between cells, so instead of a single point, a larger area, those are called belt desmosomes. And the actual protein that makes up these desmosomes between the cells, these are called cadherins. And as you'll see later along in the textbook, some types of cancers actually lose this protein, which causes cells to stick together and allows them to float off. But just a little bit of food for thought for later. And then lastly are these occluding or the tight junctions. And this is something that creates a continuous barrier and actually restricts movement of things between the cells itself. So this is actually going to exclude the movement of any kind of larger molecules or even ions themselves. And these kind of junctions can actually dissolve and reform either to allow epithelial proliferation, so the growth of more cells, or if inflammatory cells actually need to move through here, they'll dissolve and then sort of recreate after the passage of those cells. The internal cell components there are kind of some standard components that we'll see here: the nucleus, the endoplasmic reticulum, ribosomes, Golgi lysosomes, and proteasomes. Now, because each of these are contained within their own unit, there's actually individual functions within each of these organelles that don't necessarily work together. Like a proteasome being mixed around with your nucleus isn't going to end well, so they're actually sequestered in their own organelles. So some of the primary internal things within the cell: the nucleus is going to contain the cell's genetic material. The rough endoplasmic reticulum is going to produce proteins for the plasma membrane, or any proteins that are going to be going outside of the cell. The Golgi apparatus, this is also involved in the assembly of proteins, and any proteins that are not meant to be sent out of the cell, so proteins intended for the cytosol or fluid within the cell, these are generally made on the free ribosomes. The smooth endoplasmic reticulum, so this is the endoplasmic reticulum without ribosomes embedded within it. These tend to be more abundant in cells where steroid hormones and lipid synthesis is higher. And this is because these are both lipid-based products, so you'll see these more often either in the gonads or the liver. Lysosomes, these will actually degrade enzymes, often for the digestion of larger molecules. And proteasomes are specialized versions of these lysosomes, and these break up denatured proteins and release peptides. And there's also vesicles that exist within the cell itself, and these will shuttle materials around that are destined for specific sites within the cell. So they're kind of like little taxi cabs for stuff within the cell. Cellular movement is controlled by the cytoskeleton. So these are tiny little filaments within the cell itself, and this will help your cell maintain shape, maintain the organization within the cell, help the cell move around, as well as move things within the cell itself. And part of maintaining the cell shape and internal organization is important for the cell polarity. So some cells actually have a top and a bottom to them, and healthy cells will have this maintained kind of top and bottom, whereas unhealthy cells sometimes lose this polarity to them. Now, the mitochondria is the cellular site where all of the sort of energy generation occurs. Now, it's going to produce energy, or ATP, mainly from glucose and oxygen, and then it's going to give off a bit of water and CO2. Now, this pure kind of oxidative phosphorylation, it actually burns through glucose to make the energy, the CO2 and H2O, and it's pretty efficient. It's only going to give off pretty much energy, and there's not really any carbon leftovers that can be used as building blocks for anything else. So it's not going to be able to construct any of their lipids or proteins when it's generating energy. But there are certain cases where rapidly growing cells, both benign and malignant, they will increase their glucose intake and decrease the amount of ATP they're producing per glucose. And what this does is you still get a tiny bit of energy, like a fraction of what you normally get, but your cells can get access to a lot more carbon, which are sort of the building blocks for making things. And any kind of growing cells, like in a fetus, for example, it needs a lot of these building blocks to put together tissue. But malignant cells also do this because they're growing. So this is something that we see sometimes. Interestingly, the mitochondria have their own DNA as well, which is all maternally inherited. And they also play a role in cell death, which we will touch on next. So cell death can be either a normal or an abnormal part of cell life. When it is an abnormal process, it is generally due to some kind of external cell injury, like a toxin, trauma, or ischemia, and this often causes some kind of damage to the mitochondria. once the mitochondria becomes damaged or dysfunctional, it's not going to be able to generate ATP. And recall that those ion pumps need to continuously force, ions out of the cell to keep water from accumulating within it. And when this breaks down, these cells often, you'll see them start to swell, and eventually they die. But that swelling is one of the kind of early signs of, of this kind of cell injury. Now, apoptosis, on the other hand, is a more sort of natural type of cell death. It's a programmed cell death, and it's actually important for normal tissue development. And it undergoes or it occurs through both intrinsic and extrinsic pathways. So extrinsic is something acting on the cell, telling it that, it's your time, get ready for it, you're going. Whereas the intrinsic, if the cell senses that there's some kind of DNA damage that has occurred, or if there's intracellular stress that the mitochondria is involved in, it'll actually say, okay, yeah, we're no longer functional, time to pack our bags and we're done. Now, the mitochondria itself that we were just talking about, they actually contain a substance called cytochrome C, and when the mitochondria becomes damaged, the cytochrome C is actually going to leak out into the cytosol, and this is going to activate something else called a caspase, and this is an enzyme that is going to induce apoptosis. Now, as we were saying, this apoptosis is sort of a normal programmed cell death, and when it isn't occurring properly, then your cells are not dying when they're supposed to, and this is a potential cause of malignancy or cancer. And on the flip side, if there is too much apoptosis, then you're just having premature cell death. So this is sort of a finely balanced activity for your cells. Next, communication between your cells. At its most basic, cells need outside signals just to determine whether or not they actually live. Now, these signals may also tell cells to just sit there, hang out, do nothing. They may tell it to perform its sort of intended function, maintain its organization, or to respond to stress itself. Now, the cells respond to several groups of signals, which are largely classified into signals that, let's say, there's damage to nearby cells or there's pathogens present, and these cells will sense and respond to these danger signals and to invaders like microbes. There is communication that is contact with neighboring cells. So this is things like the adhesion molecules, the gap junctions, those desmosomes between cells. There's signals that tell it is in contact with the ECM. There are also signals that say there have been secreted molecules like growth molecules or inflammatory cytokines. And there are also hormones which come from various endocrine organs and can act on different cell types. So some of these extracellular signaling pathways are again divided into some further types, and this is just based on over the distance that the signal itself functions. So these paracrine signals, this is just cells that are in the immediate vicinity of the cell receiving the signal. And in order for this to occur, the cell needs to, or the signal itself needs to be rapidly degraded. It has to be taken up quickly by other cells, or it has to get trapped in the ECM and not travel very far. In autocrine signaling, the molecules that a cell secretes actually act on that same cell. So there's this is sometimes what you'll see with positive feedback loops or negative feedback loops. Endocrine signaling is when some kind of a meteor is released into the bloodstream, travels a far distance away, and then acts on a target cell that is far away from the cell that actually released that signal. And then synaptic signaling, this is when activated neurons are going to secrete a neurotransmitter across a small junction, which is a synapse, and actually act on the target cells themselves. Now, all these cell signals and signaling molecules are received by a specific protein on the receptor cell. The signaling molecules are called ligands, and these ligands will bind with a specific receptor on the cell surface, or they sometimes get brought within the receiving cells themselves. And once this signal is received by a cell, it often will activate some kind of response or process within the cell that is going to kind of alter the cell function, maybe modulate gene transcription, or basically tell it to do something different than it's already doing, or again, tell it to keep doing what it's doing. All right, now all your cells are going to exist in a space known as the extracellular matrix, or the ECM. And this is a network of interstitial proteins, and these are just proteins between things like themselves that actually is a large portion of any tissue in your body. And it functions to provide some kind of mechanical support for anchoring cells, and also offers some support for cell migration and cell polarity. It helps control cell proliferation. So the ECM actually contains a bunch of growth factors, which will often get activated if there's any kind of injury or inflammation that occurs. It acts as a scaffold for tissue renewal. So normal tissue actually requires a basement membrane to grow on and to attach to. So this is critical for tissue regeneration. And it also creates microenvironments. So the basement membrane that cells often attach to often is a boundary between things like the epithelium, so your skin, and underlying tissue. And in some organs, it's actually supportive and functional. So the kidneys, for example, it actually forms part of the filtration apparatus within your kidneys. Now there's two forms of this ECM. There's a basement membrane form, which is a pretty highly organized array, which usually forms around epithelial, endothelial, and smooth muscle cells. And even though it's called a membrane, it's still fairly porous, like things can move through it. And this is largely formed up of type 4 collagen. And you can just tell yourself type 4 is on the floor, and the basement membrane is like the floor for cells to attach to or to sit on. And there is also the second form is the interstitial matrix. So this is present in the space between all the cells, and it forms a kind of gel which is made up of different types of collagens, as well as things like fibronectin, elastin, proteoglycans, and hyaluronate. And most of this is going to provide things like tensile strength and recoil, compressive resistance and lubrication, as well as just extra binding elements to keep cells stuck to one another. So a bit more just on this interstitial matrix. The collagens themselves, they come as fibrillar or non-fibrillar collagens. These fibrillar ones, these are actually a major proportion of connective tissue in structures like bone, tendon, cartilage, blood vessels, and skin. And these are also involved in healing wounds, especially with scar tissue. And non-fibrillar collagens, these are actually the structure of the basement membrane. So this is the type 4 collagen. Again, the type 4 is on the floor, an easy way to remember what type of collagen makes up this type. The elastin that we see here, this is where you're going to get your tissue recoil and shape recovery after any kind of physical deformation. So things in your body that utilize this a lot, your cardiac valves or any kind of large blood vessels where there's a strong pulsatile flow. And this is also found in things like your skin, the uterus, and ligaments. And then lastly, these proteoglycans and hyaluronan. These are just a kind of a compressible gel that is going to give you a bit of resistance to any kind of compressive forces. And these are often found in things like joint cartilage. And this is also a place where they're going to provide a bit of lubrication against any kind of bony surfaces. Now, growth factors. These are important to help stimulate the activity of genes that are required for cell growth and division. So this is going to help actually send cells into the cell cycle. It's going to get rid of any kind of blocks on cell cycle progression, which again helps with replication. These will help prevent apoptosis, so cells kind of need a constant stimulus to say, stay alive, stay alive, stay alive, otherwise they won't. And it can also help cells create different components within themselves that are required for further division. Now this can also drive a bunch of non-growth activities, so this can tell cells to move somewhere, so migrate. This can tell cells to differentiate as well as produce some kind of product, so their synthetic capacity. And now these growth factors are involved both in the proliferation of cells in the steady state, as well as after any kind of injury. So when a cell is damaged beyond repair and it is going to die, obviously you need to replace it, so that's where these will come in. Now, uncontrolled proliferation is not good. So, too much of a good thing ends up being bad. So if these growth factors get dysregulated, or if the signaling pathways get kind of turned on all the time, this is where you'll get usually some kind of cancer development. So the growth factor pathway genes are generally called proto-oncogenes, and these are the good genes. The proto is sort of an original or early or primitive. That's what that means, and the onco is cancer. So a proto-oncogene is a very kind of primitive cancer gene, whereas a straight oncogene, that is the one that's, that will drive cancer. So the proto-oncogene, not a concern. The oncogene is the one that you're worried about. Now, some examples of common growth factors are things like vascular endothelial growth factor, and this is something that will tell your body to grow vessels. Transforming growth factor beta is another common one, and this is telling your body to make collagen and some of those other ECM components that are going to help with scar formation and fibrosis. The cell cycle itself is what cells will go through to help maintain their population. Now, the key elements in any kind of cellular proliferation are some kind of accurate DNA replication, along with a coordinated synthesis of everything else within the cell. So the endoplasmic reticulum, the Golgi, all those things, they need to be duplicated before the cell can divide. So the sequence of events that typically results in a successful cell division, it first will undergo the G1 phase, which is just a growth phase. The cell will get bigger. Within the S phase, you will see some DNA synthesis, so it's going to duplicate the DNA. G2, there is more growth, and then in the M phase is your actual mitotic phase. Cells that are not actively cycling are said to be in the G0 phase, and the cells can enter the G1 either from this kind of G0 pool, or they can jump right into G1 after completing a round of mitosis. And this is something that you'll see in cells that are continuously dividing. Now, the cell cycle itself is regulated and driven by proteins called cyclins, and having a very original name, cyclins, because they are cyclic in nature, their levels go up and down. Now, these cyclins are also controlled by cyclin-dependent kinases, or CDKs, and these are just the associated enzymes which help drive these cyclins. Now, there are also various checkpoints within this cell cycle, or this sort of growth phase, just to check for things like DNA or chromosomal damage. So, for example, there is a G1 to S checkpoint, which is actually going to monitor the integrity of the DNA before the cell commits a bunch of resources to DNA replication. So it doesn't want to replicate a DNA strand that isn't correct. Later on, within the G2 to M phase, there is a restriction point that just makes sure that the genetic material has been replicated correctly. So again, G1 to S, make sure the DNA is all right before it copies it. Then the G2 to M, there's a check to make sure that all the copied DNA was basically copied correctly. And if there's any kind of error in the DNA code, or if there's a big kind of genetic derangement that can't be repaired, the cell will either undergo apoptosis, so it'll say, it's my time, I'm signing off, or it will just enter a non-replicative state, which is called senescence. And this is a state where it's just not going to replicate. It'll just continue to sit there until it is aged out and it's time to go. Now, there are CDK, or the cyclin-dependent kinase inhibitors, which will actually enforce these cell cycle checkpoints. So the cell won't be able to progress past that point until the inhibitor is gone. But if these CDK inhibitors are defective, then cells with damaged DNA can actually proceed through this cell cycle and mutate and divide with mutated DNA, and that provides an ever increasing larger number of mutated daughter cells, and these often have the potential to become malignant tumors. All right, and stem cells. So these are cells that can give rise to a variety of different tissues. During development, there are embryonic stem cells, which basically give rise to all the differentiated tissues within your body. But the adult, or any adult, also has stem cells, and these are what's going to replace any kind of damaged cells. They're going to maintain tissue populations, as individual cells within them undergo replicative senescence. So basically they're getting too old to keep replicating. Now, for example, skin cells are continuously replicating, and they have stem cells, skin stem cells, which are continually replacing the upper layers of skin before or as these sort of age out and die and are shed. Now, there's two main properties of stem cells. They need to have some capacity for self-renewal, so their numbers need to be constantly replenished, and they also undergo asymmetric division. So when a stem cell divides, there is going to be one daughter cell that stays as a stem cell, and then one daughter cell that actually will differentiate and turn into a mature cell of whatever that cell type is going to be. Now, this first type of stem cell is the embryonic stem cell, and this is the one that will give rise to all the different cells within your body. These are the most undifferentiated, so the kind of most basic type of cell, and these ones are the ones that have a virtually limitless self-renewal capacity, and like I said, will give rise to any type of cell within your body, and this is said to be a totipotent type of cell. Then the adult stem cell is the other sort of common type of stem cell, and these are found in all the regenerating tissues within your body. So for example, the epithelial cells that are sort of in constant turnover, or your blood cells, which are constantly being produced. Now, like we said before, there is an asymmetric division. We're not seeing it here, but the stem cell at the top, as it's giving off this myeloid or this lymphoid stem cell, there is actually another stem cell that is staying just as a stem cell. So it's not gonna differentiate any further, and it's gonna make sure there's always a pool of these stem cells sitting there. And then this is generally the path that it'll follow. That one daughter cell will start to differentiate further and further down the line until you get a final functional cell that your body is actually going to use. And this is going to wrap up the Chapter 1 of Robbins review. Thanks for tuning in, and I hope to see you in the next video.